around the world, causing a pandemic that threatens global public health. To this day, it remains uncontrolled. COVID-19 is caused by Sars Cov-2, a novel beta coronavirus. Like other coronaviruses, Sars Cov-2 has 4 structural proteins, with the spike protein being absolutely essential for viral entry. Upon entering the body, Sars Cov-2 gains access to host cells through the binding of its spike protein to the host receptor ACE2. Cleavage and activation of the spike protein by host proteases enables membrane fusion, and the viral RNA is released into the cell. The host cell and its machinery are hijacked to facilitate viral replication and translation of essential proteins. Ultimately, new virions are assembled and released. Sars Cov-2 infection does not go unnoticed within the host, with innate immune sensors able to recognize viral elements such as RNA and proteins, leading to the activation of an interferon-dependent antiviral response. A characteristic of Sars Cov-2 infection is the large latitude of associated symptoms, from asymptomatic through to death. One reason may be the varying immune responses between patients, with mild cases assumed to have an effective response. However, Sars Cov-2 has evolved ways to interfere with this interferon-dependent response, causing an unbalanced and excessive pro-inflammatory response. In the more severe cases of COVID-19, this may lead to a cytokine storm and hyperinflammation with the overexpression of pro-inflammatory cytokines. One promising strategy to treat severe COVID-19 is the blocking of these inflammatory signaling pathways, using therapeutics routinely used in the clinic, such as dexamethasone. But how do we target treatment towards SARS-CoV-2? The initial strategy to fast-track a treatment was the repurposing of approved drugs, with the potential to act on the early stages of SARS-CoV-2 infection. Such drugs included the antiviral remdesivir, which targets viral replication. But after months of trials, repurposed drugs have had a limited success and have been proven to not be enough. Therefore, there remains a vital need for the development of novel therapeutics, specifically targeting SARS-CoV-2. Scientists are exploring a number of ways to disrupt the viral entry and replication cycle, using computational screening of small molecule libraries, as well as isolation and production of neutralizing antibodies from recovered patients. Furthermore, these antibodies can be used in combination, with cocktails consisting of 2 or more different spike-targeting antibodies emerging as very promising therapeutics. Looking to the future, we need a preventative strategy, and thus there is an urgent need for a vaccine against SARS-CoV-2, with it playing a significant role in curbing viral spread. Currently, scientists are exploring all the potential strategies to develop an efficient vaccine, aiming to induce neutralizing antibodies against essential viral proteins, such as the spike. The more advanced vaccine candidates moving into clinical trials include non-replicating viral vector-based vaccines, as well as novel mRNA-based vaccines. We are only at the beginning. There is a tremendous need to increase our knowledge of the SARS-CoV-2 infection cycle and the cellular recognition pathways involved, as well as the varying immune responses in COVID-19. Invivogen is assisting in the continuing global research effort on SARS-CoV-2, with the answers to these fundamental questions ultimately needed for the development of safe and effective therapeutics and vaccines.